you for inviting me to this conference. I'll be talking about inhalable powder loaded with chitosan nanoparticles for protein drug delivery. So we are particularly interested in pulmonary route for the delivery of macromolecules because we know that it has large absorptive area because of high permeability. It's highly permeable due to its high vascularity, low proteolytic activity as compared to oral routes, and we avoid the first pass effects by pulmonary delivery. It affords rapid onset of action and is an invasive administration, and of course, these characteristics will increase patient's acceptability and decrease drug bioavailability. However, as any other route of administration, pulmonary route has its own limitations like airway geometry, mucociliary clearance, and phagocytosis. So this needs the delivery or the design of an adequate drug delivery system to avoid these limitations. The general objective of this project was the development of an inhalable microscale powder based on chitosan nanoparticles for protein drug delivery, and then investigation in vivo of the capability of these nanoparticles or microspheres to deeply deposit the chitosan nanoparticles in the lung and induce a systemic effect. So we selected chitosan nanoparticles. Actually, they have been largely explored in the design of drug delivery systems because they are biocompatible. They afford drug protection and stability. They are mucoadhesive and they act as permeation enhancers and they afford also high drug loading capacity. And chitosan, we know that it's amenable to chemical modification, and this is very important for drug delivery purposes and targetability. So chitosan nanoparticles have demonstrated increase or enhanced drug bioavailability in general. So first of all, we designed or prepared our chitosan nanoparticles, and after the preparation of nanoparticles, we further transferred them into microscale powders using spray drying. So we have two processes. We have nano-encapsulation and we have micro-encapsulation. So starting with the nano-encapsulation, and we use a technique called the inotropic gelation technique, which consists of two phases. We have the anionic phase, where we have the cross-linker TPP, tripolyphosphate, which acts as a gelling agent, and we have the macromolecule of interest like insulin, which is negatively charged, and on the other side we have the cationic phase, which is chitosan. So we add the anionic phase over the cationic phase under magnetic stirring during 10 minutes, and then we have a suspension of nanoparticles which will be centrifuged to isolate the nanoparticles and get a pure chitosan nanoparticles loaded with insulin. However, if we use the nanoparticles for pulmonary administration, it's impractical. Why? Because when we have nanoparticles, we have large surface areas, so they are hyperactive and particles. We have a particle-to-particle interaction due to the surface area, so they are unstable formulations. And also, we will have problem in handling of these particles, dry powders, because exhalation of low inertia nanoparticles. So to solve this problem, our group found or developed the microencapsulation method of the nanoparticle using spray drying. So the microencapsulation, here we further load the nanoparticles in a microstructure matrix using a thermoprotector or another excipient carrier. And this technology method actually improved the aerosolization properties of the nanoparticles, the flowability of the formulation, improved powder handling, and encapsulation of the nanoparticles, further encapsulation of nanoparticles in microstructure powders, further protect the encapsulated proteins, okay? And the most importantly, microencapsulation affords proper aerodynamic diameters, which is very important for deep lung deposition. This is very important point. And to do this, we need carrier excipients, and usually people use different carrier excipients, like, for example, lipids, amino acids, and sugars. In our case, we use different sugars, like, for example, lactose, saccharose, and mannitol. But in this study, we used mannitol because of its safety record, and it enhances actually the formulation stability and aerosolization performance of the powder formulation. And we found that it maintains the biomolecule integrity upon spray drying. Okay, and also it improves or promotes drug penetration through thick mucus via osmotic and mucolytic effects. So we use mannitol for spray drying or microencapsulation of the nanoparticles. So the microencapsulation method using spray drying method and mannitol thermal excipients not working anymore. Okay, vale, gracias. So the microencapsulation very well initiated by, we get here the isolated nanoparticles, okay? And we add different concentrations of mannitol and mix them under drying at room temperature. So we have here the mixture of the nanoparticles with mannitol, and we use different concentration of mannitol to the nanoparticles. You can see here we have 30 to 70 and 20 to 80 and 10 to 90 weight ratios of nanoparticles to mannitol. And this is the spray drying technique, spray dryer, Bushy mini spray dryer. And also, of course, as we optimize the formulation parameters, we also worked on the process settings, the inlet temperature, outlet temperature, and feed rate in order to have the microspheres of interest. So here we have the microspheres or the microencapsulated nanoparticles as evidenced by the same images as seen here down. So of course we have two processes now. We have the nanoencapsulation by oxygenation. We got nanoparticles. We characterize the nanoparticles, physicochemical characterization of the nanoparticles. And after that, after the microencapsulation, we also characterize the microspheres we got. So starting with the physicochemical characterization of the nanoparticles, insulin-loaded chitosan nanoparticles, here I would like to mention that we use two different molecular weights of chitosan. We have 213 and 113 of chitosan. And we characterize the nanoparticles. Here we have 213 and 113, and we characterize them. Actually, we got a lot of formulations, and finally we screened out the formulations for the process yield, the size, the theta potential, association efficiency of the protein or loading capacity. And finally, we selected the most important, these two formulations to proceed with, which they achieved process yield between 48 to 56%, and they had nanometric size between 289 to 404 with positive theta potentials ranging from 26 to 32 with high association efficiency of the protein ranging from 75 to 83. And also here we have the morphological appearance of the nanoparticles, chitosan nanoparticles, which looks like a solid, compact structure and round. And regarding the stability of nanoparticles, we studied their stability in the buffer at 36, and we noticed a change in the size. However, that the nanoparticles, they remained in the nanometric range and it was suitable for this application. And most importantly, we look at the in vitro release of chitosan nanoparticles, and we noticed that after 15 minutes, we have a burst release. We have 50% of the insulin release, and after that, we have a kind of sustained release. We got 80% of the protein release within 2 hours. So we have here 50% of the protein release in best kind of burst release, and this indicates that we start to assume that we might have a proportion of insulin dissolved at the surface of the nanoparticles, and another proportion might be encapsulated in the core. So to make sure of this, And this also indicates weak binding, actually. So to make sure of this, we conducted further studies here, and we conducted surface elemental analysis where we used X-ray photoelectron spectroscopy to check if we have a proportion of insulin adsorbed on the surface of the nanoparticles, which is responsible of the first the burst release. And here we use pure insulin powder, blank chitosan nanoparticles, and also insulin-loaded chitosan nanoparticles. We can see that the pure here in the blank chitosan nanoparticles, we didn't have any signal for sulfur and zinc, which is attributed to insulin. However, we have similarity in the surface characteristics of pure powder insulin and the insulin loaded chitosan nanoparticles. And if we look to the signals of sodium and phosphor, we have they were available in the surfaces of both blank chitosan nanoparticles and insulin-loaded chitosan nanoparticles. However, if we look at the atomic ratios, like sulfur to carbon and oxygen to carbon and sulfur to nitrogen, we have a kind of the ratios here, they are double or more than the ratios of the signals appeared on insulin-loaded loaded chitosan nanoparticles. This means that we have similarities on both pure insulin, the surface of pure insulin and insulin-loaded chitosan particles. However, the ratios were higher in the case of insulin, and this tells us that we have a proportion of insulin adsorbed on the surface of chitosan nanoparticles. Not all of insulin, but proportion, because here we have the ratios, they are higher in the case of pure powder of insulin. To make sure of this means that we have weak binding, why we have a proportion adsorbed in the surface. So we conducted another experiment. Actually, we conducted a lot of experiments. However, this was a nice experiment which told us about the association efficiency of binding constant of insulin to the protein. And this is a proton MR study. What we did in this experiment, we did a titration study between insulin, different concentration of insulin and blank chitosan nanoparticles, and we checked for chemical shifts of the interaction between of the proton signal between chitosan and blank chitosan nanoparticles and insulin, and we noticed that the proton signal changed, and the maximum change was at 3.78 ppm. And finally, we fitted the data between chemical shifts and insulin concentration, and we could, using relevant equations, calculate the binding affinity constant of the insulin to the nanoparticles, and found it about 19,185 molar, and it is actually weak. It reflects weak binding of insulin to the nanoparticles. Then regarding the physicochemical characterization of the inhalable powders or microencapsulated chitosan powders. Of course, we have two powder formulations containing chitosan nanoparticles loaded with insulin and with 2 molecular weights of insulin. And we characterize the powders first for their diameter, Ferris diameter, and aerodynamic diameter, and real density. Most importantly, the aerodynamic diameter was between 2.5 and 283, and this is very important. Actually, if we need to deposit the powders or inhalable powders into the deep lung, the aerodynamic diameter should be between 1 to 5 microns, and actually their aerodynamic diameters was in the desired range. And here we have the SIM micrograph of the microspheres or the powders, which have the spherical morphology for both formulations. And again, we wanted to know if after spray drying or microencapsulation of the nanoparticles, we want to make sure if the nanoparticles were entirely and homogeneously distributed throughout the mannitol matrix after spray drying, and if we don't have or have some nanoparticles adsorbed in the surface. So we did two kinds of study. We did surface analysis and elemental analysis of the surface of the resultant microspheres. So we're starting with the surface structure analysis using confocal microscope. Here, and we first labeled our chitosan nanoparticles with fluorescent label. which appears in green signal, and we also labeled the thermoprotectant, which is mannitol with body buy, and we formulated the nanoparticles, including insulin, okay, and then we micro-encapsulated them using the spray drying and the labeled mannitol. Here in the figure A, we can see that the green signal represents the nanoparticles distributed throughout the mannitol matrix homogeneously without detecting aggregates or clusters of the nanoparticles. And here, figure B represents a well-defined shell of the thermoprotectant mannitol. And if we look or visualize them in the overlap channels, we can see that how the nanoparticles, they are actually surrounded with a well-defined coat or shell of the thermoprotectant, and this is nice because this is our scope to get the nanoparticles only encapsulated in the interior of the or throughout the matrix, and not adsorbed at the surface, because they will damage the protein. Okay, to make sure this, we did further optical sections of these microspheres along the z-axis. And actually, we noticed the same trend. Always, these actually the optical sections separated by 400 nanometer, and always we got, of course, from the top to the bottom. Always we get the same trend. The nanoparticles always in the center without detecting aggregates, and we have the mannitol or thermoprotected shell always surrounding these nanoparticles. This type of study, it tells us about the external and internal rearrangement of the nanoparticles and the thermoprotector in the microsphere powder. But it doesn't tell us exactly or accurately if the surfaces still have some nanoparticles absorbed there with insulin there. So we further conducted surface elemental analysis using XPS and using TOFSEM techniques. So starting with the XPS analysis, we use, of course, chitosan nanoparticles without being microencapsulated with mannitol, blank mannitol. Mannitol was, we used mannitol microencapsulated but without the nanoparticles. And we used our formulations, which they are microencapsulated chitosan nanoparticles. And if we look at the atomic composition and atomic ratios, we could find that the surfaces of the blank mannitol and the mannitol microencapsulated chitosan nanoparticles, they have the same atomic composition, surface atomic composition with the same proportions or concentrations. This means that we have only mannitol on the surface of the nanoparticles. While in the chitosan nanoparticles without being encapsulated, they have different elements on the surface and different atomic ratios. So this is another clue that we have only mannitol there. We don't have nanoparticles adsorbed on the surface. Performing another or using another technique, which is TOFSEM, time-of-flight secondary ion mass spectroscopy. Again, screening in this region, we could see the spectra of mannitol microspheres and microencapsulated nanoparticles. We got the same signals. With this signal, actually, oh my God, actually this. Okay, we could detect molecular ion of mannitol in both spectra, the mannitol microspheres and the blank ones, and the microencapsulated chitosan nanoparticles, while we didn't detect this signal in the case of non-microencapsulated or chitosan nanoparticles without being microencapsulated. However, we detected another fragment, another signal for another fragment, which was not detected in the other spectra, the powder spectra. Again, screening in another region, we could here, in the case, in the spectra of chitosan nanoparticles, detect PO3, which is attributed to the cross-linker available in the nanoparticles, which was not detected in the spectra of the powder formulations, okay? However, they were, the spectra of the powder formulations were similar. We could detect the same fragments there. And scanning in another region, again, we have the same tendency, similar spectra for the blank mannitol microsphere and the microencapsulated chitosan nanoparticles. And again, chitosan nanoparticles alone without being encapsulated, we have different signals for different fragments. For example, these fragments, they contain an A-sodium, which is attributed to the cross-linker. So this is another or further clue that the surface composition of the mannitol microsphere, the mannitol microsphere containing the nanoparticles, they had the same composition, they have the same elemental elements and with the same proportions. So this strengthens our assumption that we only have in the surface mannitol, we don't have nanoparticles. This tells or this suggests that mannitol acts as drying stabilizer, where it absorbs at the air-liquid interface during spray drying, leaving the nanoparticles inside the microsphere matrix. And this is actually refers to or indicates the success of the microencapsulation method using spray drying, using the conditions that we optimized conditions that we used in our experiment. And also this is very important because we want the nanoparticle to be homogeneously distributed and maintained in the mannitol shell, and this is important for the protection of the protein. And we conducted analysis to make sure if the integrity of the protein was preserved, and it was preserved. Okay, then we move to the in vivo study and to check if the formulations were biologically active. We actually used Harvard ventilator, which simulates the breathing pattern, physiological breathing pattern in rats. For the control formulation, liquid formulations, we used intratracheal instillation using a syringe. And here we have the formulations we used. We used the two formulations with the encapsulating powder formulation, encapsulating chitosan nanoparticles with 2 molecular weights of chitosan. The controls, we have the dry powder of plain chitosan nanoparticles, of course, without the protein. We have dry powder of insulin and we have insulin solution and chitosan nanoparticle suspension without being microencapsulated. Here we have the after the in vivo study. Here we have the hypoglycemic profile. We can see that the control formulations didn't induce or the powder formulations induce a significantly higher hypoglycemic effect, which was prolonged over 6 hours. 6 hours. Here we have the microencapsulated chitosan nanoparticles with 2 different molecular weights of chitosan. Here we have the control formulations, so we have significant hypoglycemic effect. However, this hypoglycemic effect was prolonged and more pronounced for the microencapsulated chitosan nanoparticles with a molecular weight of 113. So, and if we looked at the area under the care, again, we have the microencapsulated chitosan nanoparticles with the higher area under the care being the best for the ones which contain the chitosan of the molecular weight 113. Again, we wanted to check in vivo. Actually, the previous experiments in vivo tells us that the powders could deposit the nanoparticles in the deep lung and reach the systemic circulation and induce a pharmacological effect. But of course, we wanted further to check the deposition in vivo of the nanoparticles. We conducted confocal microscopy, actually, of course, after using labeled nanoparticles. And after 1 hour of installation of the dry powder, we expose them, the alveoli, we fix them, we slice them, and we visualize them under the microscope. And we can see from the green signal, which refers to the fluorescently labeled chitosan nanoparticles, that the nanoparticles reach the alveoli, which is important to induce a systemic effects, with a tendency to be captured in the macrophages. Here we have macrophages, alveolar macrophages here, and these actually sections for the alveoli. This is very important that our system succeeded to deposit the nanoparticles into the deep lung with ability to induce a pharmacological effect. And finally, we can see that in vivo results obtained following intratracheal administration in rats demonstrate that the developed inhalable microscale powders based on microencapsulation of chitosan nanoparticles by spray drying constitute an adequate carrier for deep lung deposition or delivery of protein drugs, systemic absorption of protein drugs. The developed also, we can see that the developed micro-nanostructure delivery system holds promise for non-invasive administration of macromolecular drugs. And finally, I would like to greatly thank my PhD supervisor, Carmen Rimonia Lopez, from NanoBioPhar Group in Santiago de Compostela, Spain. And thank you for your attention.
Yeah, thank you very much for the fast talk. And in the interest of time, I would like to give you your certificate right now, and I think you will be available for the next coffee break to answer any questions. Thank you again.